So in writing a lab report, we're going to do 3, and you'll see the first one's pretty easy, and they get a little bit harder. And so our goal will be to make them progressively better as we go through the course. And then you'll also notice that they're worth a lot of points. So most of the points for the course are about the lab report. And so you just want to take time to do a good job, but you'll see it's not a lot of work to do the lab report. And so you'll be surprised at this first one where you're like, wow, am I really done? And the answer is, yeah, if you get everything set, you're really done. And the reason that it seems like that is because the lab report should be very concise. So you don't have any kind of page limits. Short is good. You want to get to the point and have it be done. But you also want it to be complete. So you want to cover all the territory that's described in the lab manual in Unit 4. And since we didn't get to all of that in class, we'll talk through it. So you want it concise, complete, and here's why. Scientists do two kinds of writing. One kind of writing scientists do is they write up their discoveries and they publish them in journals. The other kind of writing scientists do is they have a daily lab notebook, and they just maintain that, kind of like a diary, personal journal that they're maintaining every day. And the way we talk about lab reports in this class is focused on the daily journal. And so when you're writing a lab report, you should imagine that. When you've had other science courses, your teachers or professors would have been focused on the idea of the publication. And that's, like sort of imitating the publication. And that's really foolish, because those are really products of years and years of work and just painstaking picking of words and revision and so forth. It's just not realistic. And so you want to be careful in future courses, in science courses, your professors will ask you to write up that type of lab report. This lab report will be helpful because this lab report is the basis for those larger lab reports, and it's good, and it's a better perspective and attitude. And so we want to imagine that. And here's what you imagine. You imagine that you're writing this down every day, and you're writing it down so you can remember it, and you can go back to it. So you're creating a reference document of your activities, and that maybe in like 2 years, you'll want to look back and find this. And so that's why the lab report should be concise, because you want it to be a thing that you can just flip through. And then also it should have this kind of, so here's an example of one page of a previous lab report. And you don't need to be able to read it, you can just see the format. And the format is kind of outline-like, divided into section headings. And the reason that is because you would be scanning when you're looking for stuff. So you're creating it, and of course we're not creating that many documents in this course, but imagine that you're creating one of these every day, and you're creating a huge book of them that you'd look back through. And sometimes you'd be looking back to find a result. So you're like, how did that work? I don't remember how that worked. Sometimes you'd be looking back to find a method. Sometimes you'd be looking back to find an idea. And so scientists break their writing up into these sections like that. So they're outline format, and that just allows people to scan through it. And that outline section format even carries over to the publication. So that's why writing this type of lab report would help you in any course, because they're always broken up into these kind of sections. And so here are the sections that we'll have for our lab report. And the first one will be a title. And our lab manual has titles, but our lab manual's titles are stupid. And because what you're thinking about is, again, this scanning and searching. So the title should have key words in it. Like I imagine your title will have the names of those different colorimetric assays, Lugol's, Benedict's, Bayer's. You can have all 3 of them in there because you might be looking for that, like that. And, maybe identification of biological molecules by these methods, like that. Maybe something about a colorimetric assay, like that. So it's a meaningful title. you know, not a kind of a cute title, just functional, boom, 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 like that, and it would be about keywords. Then there's a section called objectives. And objectives are kind of like an extended title, and they'd just be like maybe 3 sentences where you're just like, what are we actually trying to do today? And in the format, these would be offset, like I showed in that, where you'd have like a section heading, and you want sort of this open format that you can scan through. You can create the lab report either by hand or on the computer, doesn't matter. But you want to have it be laid out like this. Then the next section is background. And background, or you could call it the intro. And that's new stuff, new knowledge you learned you might forget. So, for example, in this, you know, it makes sense that you would have, in the background, you would list what the different assays detect, because you just learned that, and you would forget that. What does Lugol's detect? Like that. So that would be a logical thing to put in the background. But something like you would not put in the background, something like, well, water is wet. Because that's not new knowledge, and that's not something you're ever going to forget, like that. And so the idea of background is like that. Now, consequently, everybody will have different background, because, you know, some of you will have more or less experience with these, so you'll have difference. And I'm going to be judging and grading you based on what I think makes sense. But, you know, you're writing this not to please me, really, but to imagine you're creating this document that would be good for your own use, like that. So I acknowledge that that makes it a little kind of tricky, but we're going to focus on that. So you're not writing a lab report that has that's full of like the right answers and exactly the right format that I insist on, but one that you're imagining would work well. And so maybe you'll come up with some format and I'll criticize it, but you'll say, oh, isn't this a good idea? And I'll say, oh yeah, that is a good idea. I didn't think of it that way. Like that. And so the background is, there's no length to it. It might be really short. It might be kind of bulleted, you know, or it might be more narrative, full sentences and paragraphs, your choice. Then there'll be a methods section, and in other classes, the teachers will want you to write out the methods. But that would be a mistake here, because we already have our methods written out well, and we don't need to repeat them. That would be a waste of time and a waste of space. And so so what's here is, this student has just said, for the methods, it said, you know, see pages so-and-so to so-and-so in the BIO106K lab manual. So don't write out the methods, you know, because we have them. They're already really well written. And that's a good way to think about being concise, is we can just point to the lab manual and we're done. So that's simple. The methods are simple. Here's an addition to the methods is, if we did any kind of modification, you need to have some notes. So if there were some changes that you made to the methods, you want to put a section of notes, just bringing those comments up. Because if you ever did the experiment again, you'd want to see that in the methods section. So one thing that notes might have will be any modification. Another thing the notes would have is, if you made a big mistake, you might put a little note in that says, caution, don't overheat the samples. Because if you repeated this experiment, you wouldn't want to make that mistake again. Another note you might have is, if there was something dangerous, you know, in the procedure, you would want, note, you know, be careful with the such and such. Now, maybe we'll have some notes and maybe not, depending on how things went, like that. A note could also be, if you didn't make a mistake, but you heard other groups making that mistake, you could make a note of that. You know, you didn't make the mistake, but be careful with this, you know, like that. And it's just, imagine that 2 years from now, you look back, you find the method, you're going to repeat the method. You haven't done it in 2 years, And you'd want to just notify yourself of dangers and modifications and errors that were made before. And that's the place to do it. And in my personal life, I'm just really sloppy. But this is all this really neatnik stuff, where you want to put the stuff in the right section. And it's a logical thing. So anything like that you want to put in this section. So that's that. Then the next section is the results. And the results should have, there should be these two parts to the results. And one part is what I call the narrative. And that's describing your results in sentences and paragraphs. So we analyzed these samples and we found color changes of a variety of different colors when we looked at different colorimetric assays. And as you describe your results, you refer to the data. So you say, oh, see table 1, and then see figure 1, like that. So that's one part of the results. The other part of the results is the data. And in our first lab report, the data is going to look like this, where we're going to have these tables with data in it like this, where one tube turned light blue, like that. So there's going to be a descriptive table like that. And you should have a good title for every table. And so this title would be Table 1, something, something, something. And the title would be some meaningful keyword. And the idea is you want the table or any figure in any lab report so it can kind of stand alone. Because imagine, again, you're flipping through your book a couple years later, and when you flip to a figure or a table or graph, you want to be able to look at it and understand what happened there. So it kind of stands alone, like that. If you're confused, you should be able to look to that narrative and get more understanding. But the table or figure should be able to stand alone. And I described this. I said, oh, this is what I would call our raw data. And that's great, but we definitely should include a second table where we analyze that data and we change it from descriptions to what we interpret to be really positive reactions and negative reactions. Because the point of those dyes is they should be able to detect certain molecules. And so you should look through your data and look at the descriptions of the assay reactions in the lab manual and decide now which of these were positive and which were negative. And because that's really the goal of this. So you'd have that. So there's that piece of data like that. And there's this second part that we didn't get to. It's the same thing, where there's a table with raw data and then a table with our analyzed data, like that. And that's all the data we have for this. So that's one reason this is a pretty straightforward lab report. And but the key thing is to look at that. And we kind of started out in this course thinking about graphs and looking at them and thinking about them. And it's like it's logical. It's not fancy thinking. It's logical thinking. But just think about it and say, oh, this makes sense. I've made it this way because it makes sense. That's what this is all about. So there's that section, results. And then the next section will be conclusions. And those are that's just really short. It's like 3 sentences also, kind of like the objectives where you just go boom, boom, boom. This is what we conclude. It's really short and to the point. And then the last section is the discussion section. And that's a narrative. That means sentences and paragraphs. And we'll see that that could easily be a page long, just to give you a feeling for it. You could easily write a page about this kind of stuff. And the discussion is issues that have come up. Like, here's a famous thing where, let's say that you got your results and you compared to the other groups and they didn't match up. So in your results section, you just present that fact. You know, we did ours and we compared ours to others, they didn't match up. Then in the discussion section, you discuss why were results different? And the scientific logic just carries through here. Well, what would be some reasoning? If your results didn't work, what are some things? And you wouldn't want to go with the, because we're losers, like that. You instead think of something like, what are some things that could go wrong? What do you know about what we were doing that could explain it? Like perhaps we forgot to heat our samples. That was a really important step, and if we didn't heat them, maybe they wouldn't change colors, like that. So one of the things in the discussion is this, why were the results different? Another thing in the discussion can be, why were the results the same? So if you compare them with other groups and you got similar results, that's significant. We don't know what the right answer is, but if we see other people getting a comparable result, we start thinking, oh, that's probably what is expected. And you talk about that there. Looks like this is pretty similar results, and so we can start kind of concluding that milk has protein in it, like that. That's what all the evidence points to. It's pretty consistent with that. And in that context, you can say, you can talk about the idea that this is a good method because these are reproducible. So that's a key thing in science is, well, somebody else should be able to do it and get the same result. And you can write about that in the discussion. Is this a convincing assay? And if it's a convincing assay or not convincing assay, you can discuss that. What makes it convincing? Or how could you improve it? So, is there something you could do to improve the method to make it more convincing? Like that. Another thing that can come up in the discussion is, do you think these results are generalizable? So we tested it on starch. Do you think it would be true for other sources of plant material? Like that, and if so, why do you think they're generalizable? Because that's a big thing in science. You test a specific thing, but do you think that's true for stuff in general? Generalizable to different conditions, different sources of material, things like that. And then another thing in the discussion is, that's a really famous thing, is what happens next? And this is a thing where you kind of have to fake it, because you have to sort of fake that you found this really interesting and compelling and you want to do something more, like you want to test it on your cat, like that. But that's what scientists are always doing, is what are they going to do next? And that's a big part of what the discussion section is. These are found, discussion sections are found in published research articles too, where they've got their data, here's your data, they analyze their data, and they say, now here's what we think is going on. Here are the implications, here's where we're heading. Like that, cure for cancer is where we're heading, like that. And so that's a famous thing to think about. What happens next might be something new, like try it on something else, or it might be try to improve the method and test that out. I'd like to do this again, but I'd like to do it at a lower pH, just because I'm thinking that might make the results more consistent, like that. And so what happens next is that. Then here's another thing you can go to if you run out of ideas for discussion, is those questions for thought that we do. Those are actually they're always kind of like discussion questions. What about this? What about that? They're kind of points for discussion. And you don't want to just answer them. So in our regular homework, we just crank through and answer them. Instead, what you want to look at is look at the questions for thought and see if there are ideas, concepts, terms that come up there that you could put into your discussion. Either in the question itself, just the question, or in how you would answer it. And you don't have to do all the questions for thought. Just they're in Lab 4, they're just an opportunity for ideas to help you. Because your discussion should always be a narrative. It shouldn't be a list of the answers you have for the question for thought, but just use whichever ideas you can from the question for thought and plug those in, like that. And so those are some of the ideas that you could use for the discussion. And that's it for the report. Again, we're really focused on the idea of creating a daily lab notebook journal, and that's what we're emulating. And believe it or not, good scientists actually have this kind of format in their daily notebook. So, a lot of times you'll see scientists working and they'll be jotting stuff down on pieces of paper towel and stuff like that. But hopefully they go back to something like this and write it out well in a kind of laid out format like that, where they have a sort of a beginning, middle, and end of their day like that. And so that's what we're doing. And although most other teachers, professors would want you to write like you're emulating a publication, this is really the sort of the foundational skeleton or framework for all those publications too. So all these ideas are really good. The idea of there's really no room for any BS. Just go right to the point, concise. But you also want to be complete, and you want to have it be well organized, sensible, logical layout, like that. So it's not that bad. Here's just another figure I got from a student's paper, just with some data like that from one of their labs. It's a different lab report, but just well labeled. And again, that's the kind of stuff that generally I'm like, who cares? We all know what happened. But in grading this, you'll see my comments are always these really picky things, like, well, you should number your tables, so you can identify them. And so that's that. Do people have questions about the format for that?